Hello everyone, welcome to today's video, and today we will be understanding an important tool that I've been using recently, which is SAMSON. What is SAMSON? In this video we'll try to understand that. What are the different features that are available for you through SAMSON? What you can do with SAMSON if you're a biologist, if you're a computer scientist, chemist or a physicist. There are so many options that are available in SAMSON, and it's recently been updated. Also, a lot of new features are also available. So I've been using this tool, and I really like using this tool because there are so many features that are so useful in my research. Okay, so I'm going to show you all those things one by one. And after this, after we visit entire website of SAMSON, we'll move on to the actual SAMSON tutorial part, where I'll be showing you, just a taste of the SAMSON, how you can see compound, how you can, do basic visualization. And in future, what I'll be doing is making more videos on specific part of the SAMSON, because that is how we're going to learn. As you can see on the screen also, there are so many snapshots, small shots in this video that you can see. You can do a lot of things with molecules, small molecules, proteins, and nanotubes. There are no limitations as far as physics is concerned, as far as biology is concerned, as far as chemistry is concerned. You can do, and also you can analyze MD simulations. Gromix is also integrated. Autodoc Vina is also integrated. And visualization is also available, right? There are so many features. I mean, they are trying to incorporate VR also in that one. So for teaching, it's also amazing. So there are so many things that we'll be covering. So we'll start with the first tab. So this is the SAMSON website. What you can do is just type SAMSON Connect, and then you will be directed to the SAMSON website. Okay, let me show this, right? So as you know that we do everything from scratch. So we'll start with SAMSON Connect. This is what you will be doing, and as you can see, this is the company name, which is One Armstrong, and then SAMSON is the software that was developed by this. Okay, so we can directly go to the first option that is available, SAMSON Connect Home, and that is where I was, right? So this is the interface, and in the interface you have multiple options. I'll just close the first tab down, and you can see another message got popped up, which is get unlimited access for your group, department, or institution for an academic site license. So if you are working in a lab which is in the academic institution, so you should definitely apply for this academic site license, where with a minimum cost, I think you can get a lot of features. I'll tell you about what are the features that are available in the basic version, which is free to all. SAMSON also provides that, right? We'll talk about that, but let's scroll down and see what are the things that are available to us. So here you can see you have to register to SAMSON, and then that software that you're using will be customized based on what are the things that you're adding in SAMSON. So as you can see, video is available. When you see the video, the rendering quality is pretty amazing. It's awesome, and it can create the visualization which are really, really professional. And interface is also great. I'll show you later on. Now, if you go down, you can see this is the interface, and there are some windows that are available right here. I'm not going to show you what are these different windows, because that is the part where we'll cover in our tutorials. So here you can see SAMSON is growing pretty quickly. A lot of people are working with SAMSON. A lot of good researchers, they are working with SAMSON. This is the overall map of people who are working with SAMSON, and you can see everywhere, everyone is surely using SAMSON throughout the world. So this is the data. So this is the data that is shown on their website. Obviously, this is the data that is going to change. Hopefully, after making this video, this will increase, and you guys will use SAMSON, because I recommend you to use SAMSON. Modern feature packed platform for molecular design, that is really, really true, because you have a lot of features like your AI assistant is there. So let's just imagine you have ChatGPT integrated into a molecular visualization tool that will help you. If you have any query, if you have any doubt, there is no need to go anywhere to read documentation. It is available to you. It is available in a nice, small window where you can type thing, and immediately you will get the information that you need. Okay. What else? You can build and simulate at the same time. There are features that are available. You can create the molecules. So probably what you are seeing right over here is creation of a molecule. So you can also do and create your molecule. There is no need to go to another software and create your target molecule that you want to explore, right? Next, control. You have a lot of control over your attributes that are available. And next you have, as you can see, visualization is pretty awesome. You have various tools that are available to visualize the molecule in a different types of modes that are available. For biology, look at this. The images are so great, and you also have video editor, I guess, platform for science communication. So here you can create awesome videos that you can share with your during your conferences, your presentation, especially when you want to present something. If you have a nice video, that will make a long-lasting impression, and then whatever you show is secondary, because you have made that impression by creating an amazing visualization. And SAMSON is pretty amazing as far as the visualization is concerned. You can integrate trajectories and create them. As you can see, how it's been built from one trajectory to another one. And then there are additional camera features. There are so many features. I don't know how many I will be covering. Rendering is also available. Look at this. You have a light in the center, and then everything else is illuminating on the sides. Wow. And then these are some of the additional features, extensions that are available in the marketplace. We'll explore the marketplace also. Then I'll show you what are the features available. Look at this. In the background, what you are seeing is you can create by using SAMSON. And if you are a researcher, you want your research on the cover page of any journal, they want professional looking images, and SAMSON can provide you that. Beautiful images with a lot of high quality rendering. Look at this. What is this? Help included. So you also have a lot of help from SAMSON that you can follow and learn those things. Find everything. You have a tool to find various things that you are not aware of. SAMSON will talk to you. SAMSON will help you out. There is no need to go anywhere else. And if you don't want to watch any video, SAMSON itself will provide you a lot of help, and then you should be doing all those things. And if you need any help, I'm here, right? I'll help you. I'll be creating more videos so you can learn with me, and then use SAMSON for your research, for your computational chemistry or computational biology or physics, whatever you're doing. Okay, so we'll start with the platform. That is, I think we have already covered. I just want to click on this. Yeah, that is what we have covered. Here is the tab where you have to register to this. And then let's go to the pricing. I have already registered, so I'll show you after registration what are the things that you can get. Okay. Next is pricing. In pricing, you can select your plan. So you have starter package, professional package, enterprise package also. So as you can see in the starter one, it's free forever, right? There is no limitation. And key features that are available, a lot of features are available on the starter plan. Look at this. AI assistant is available. Physically based modeling, you can do interactive simulation, you can do cloud computing, you can do publication quality visualization, you can do template based animation is also free. Universal file embedding is free. Integrating Python development if you want to develop something, so that is also available. ChatGPT 3.5 based AI is available in this case, not 4 is available in case of the professional plan. One free extension is also available, so probably if you want to do Autodoc Vina, so that is available. 1 GB for public and private documents. So storage facility is also available if you're not using your own system. Cloud computing is also available for you for basic functioning. That is free. So you have to just register to this, get all the features, and then start working with SAMSON, especially when you are a researcher and you are working with protein visualization or small molecule visualization. This is going to help you a lot. Next one is professional. In professional, you have to pay marginal money like $10.84 US per month, right? This is for academic. So for academic, the plan is really cost efficient. Commercial 109.38 per month. That is if you are working, if you're a company or something that is not related with academic research, then commercial plan is for you. And as you can see, a lot of features are available in the key plan. But you have dark mode because that is something we all like. We have AI voice control, and then you have ChatGPT 4 based AI. So AI is more intelligent in this case. You're gonna get help from AI very fast. Unlimited movie length. You can create long movies, right? And they are really good, as I told you that. Okay, everything in professional plus. So everything that is available in the professional plus, which you have to talk to these people, request demo on that one. Okay, but I think as far as if you are serious researcher, if you want to do a great research work, so it's time for you to move to SAMSON and then start doing it. Free plan includes a lot of things. So definitely what you need to do, I'll tell you. Okay, there are some frequently asked questions. I mean, you can go through these questions. They are pretty straightforward. Can I use SAMSON for free? Yes, SAMSON is free to use for everyone, which is great. And then you can see it's free of cost, but you have to upgrade the plan for premium features. Can I cancel my plan? Yeah, absolutely. You can cancel it like Netflix. You can cancel anytime, right? Can I switch plan after I start a plan? Yes, you can just change. You have to sign in, and it will create a profile in SAMSON, and then you should be able to use whatever you want to use. And there are so many questions. If you have any doubt, just let me know. I'll make another video on this. But this video, I'm creating a little bit more detailed video because this will be the first video on SAMSON. A lot of people are going to watch this video, and I want this video to have everything, at least the introduction of most of the thing, and then we will break it down on different aspects one by one. Okay, so as you can see, pricing is this. Marketplace is where you will buy those extensions, and those extensions, they are going to automatically added into your SAMSON platform. So you can see you have the most of the feature extension will be Autodoc Vina. A lot of people are doing molecular docking with proteins. So Autodoc Vina is there, and then you can do molecular docking with not only single molecule, but library of the molecules very, very quickly. SAMSON is going to help you out in that. Molecular cycles you can do, NMR2, and all these extensions are available, and then we can create smaller videos on each of these extensions, how we can use them, and what are the output that you get from these. Then for life sciences, these are the extensions. So many extensions. GrowMax is also there. Pretty amazing, right? Because when you perform GrowMax, you need to create your own analysis pipeline, and that takes a lot of time. If you have created, that is fine, but you should explore, because in this one you can get a lot of features, and then there are so many other features, so many of them, if you're working in life science. And then physical material sciences, also a lot of features are available. Computational chemistry, chemical sciences. If you want to create a force field, that is also available. So many extensions are available. And in near future, you can expect a lot of other extensions are going to come. Now, last thing is resources. Resources tab, if you click on this one, you can see multiple options are available, how many computational credits you have. That means if you want to use the cloud-based facility, you need to have credits, and then you can purchase the credits, and then you can use this. Get free credits is also available. I don't know what is that. Let's explore. I don't know. Get free computing credit by referring friends, obviously. Yeah, right. So you can sign in, you get the code, tell others to use the SAMSON, and then slowly you can have more credits. For example, when your friend purchases 100 computing credits, your referral code, we give them 10 extra credits for free, and we'll give you 2 computing credits for free. So these are really helpful, because you can do a lot of computational. For example, if you want to do molecular dynamic simulation, you need a system. So that credit is going to help you in performing cloud computing, which is great. Okay, what else? In resources, documentation is also available, and we have the AI to help us with the documentation, so we should be good. Discussion forum is also there. Blog is also there, and if you click right over here, it will direct you to the forum, and if you click on the blog, it will direct you to the blog one. And as you can see, general discussion is going on. There are so many posts, there are so many topics. We will get back to this one later on, right? We need to do some action. Okay, so tutorials are also there. So there are so many different tutorials that you can follow along, and then understand whatever you are interested in. For example, I'm more interested in visualization, Vina, GrowMax, protein simulation, and also I'll be doing more or working on that aspect of SAMSON. Okay, blog is done. So I think we have covered pretty much everything on SAMSON. Now I'll show you, when you sign in, what are the options that are available and how you can proceed. Okay, so I have logged in. Logging in is pretty simple. You have to just give your email ID, and then you have to enter the password, and you are now inside SAMSON in your profile. As you can see, this is my profile, and I can show you. When you click on this one, your profile will be shown to you. You have everything right over here. All the links are available, and it will ask you for having those links. Next is, you can click on this tab, download, and when you click on download, what will happen that another window will open up where you can download SAMSON. Pretty straightforward. Okay, when you click on this one, a file will be downloaded, save file will be downloaded, and then you can just install the software. All right, now let's go to SAMSON and understand various tools that are available and how you can use SAMSON from scratch. And then I'll be doing few things, not all of them, because this video is going to be very, very long. Okay. I want you to download this and also, follow along with me, because that is the best way to learn things. And then initially you are going to have some issues with the mouse button and all. I'll help you out with that one. So let's start the tutorial and let's see what SAMSON provides you. All right, everyone. So when you download SAMSON, after you have this icon available, just double click on this one, and then the SAMSON 2024 version, which is the most latest version right now I'm using. Slowly it's going to get updated and we'll have more features. So this is the interface of SAMSON. It looks pretty nice, and you can customize this entire interface. As you can see, the theme is pretty amazing. It looks very nice. Visually, it's very appealing. Everything that is available in SAMSON, as you can see, a lot of effort is being added by the team. And okay, so before showing you this one, I would like to show you something else, which is the YouTube channel of SAMSON itself. So this is the YouTube channel of SAMSON Connect, where you can see a lot of videos are available, how you can use SAMSON. Okay, so there are various tools available. I recommend watching this video because a lot of those things are already covered that I've already told you that I'll be making small videos, right? So SAMSON, if you are rushing through your projects, you can watch all the videos, and everything is available on SAMSON. There are playlists are also available on various aspects. Webinars are also available. Various things are available on SAMSON. So from here you can subscribe. So you can subscribe to the channel, SAMSON channel, so that you don't miss anything. And then you can also press the bell icon, which is if you can make it to all. So you'll get all the notification on your YouTube channel, or you can get, I guess, email notifications also. So SAMSON Connect is a great tool. Okay, so let's get back to this, and this is the interface. I will show you, as I told you, I'll show you a few things that will get you started. That is very, very important in this case, right? So SAMSON is what? SAMSON, the full name of SAMSON is Software for Adaptive Modeling and Simulation of Nanosystems. So any nanosystem that you want to explore, SAMSON is going to help you. So initially you'll see a lot of options. Do not get overwhelmed with these options. So what I'm going to tell you one by one, you can just try them out. And once you get familiar with all the options, that you'll find very, very easy to use SAMSON, right? So we'll start with the first important help that you get from SAMSON, which is Assistant. We obviously have this SAMSON AI as assistant, and then it will help you to go through various things. So for example, initially what I want to do is I want to know how I can import the structure from PDB, right? So I'll give you just small example. I'll just type right over here how I can get any PDB structure in SAMSON. So this is my question, and AI is going to specifically help me what are the options available to me and how you can get the structure. So as you can see, install the protein structure downloader extension from SAMSON Connect. So a few things you need to add, and I have already added some of the extensions. They are already available. So what you have to do is you have to either you can just close this one out to see the entire interface, and you can go to apps. So this is the second important aspect in SAMSON where you need to have those apps or extensions installed. So if I click on this one, I'll get that I have 11 extensions that I've added, and then you can see Fetch is there. So Fetch is the extension that will help us to get a particular protein from PDB directly, right? No coding is required if you are not familiar with coding, so you can directly click on this extension, and then another window will be opened, and from here you just type the name of the protein that you are interested in. In this case, I will go for 4UV0. This is one of the protein structure that I work with all the time. And as you can see, immediately I got the structure in an interesting kind of visualization mode. You can see over here, right? So this is how you can get the structure. Pretty easy, right? You're not doing anything, and then you can just close this one out. And it will also give you messages that what you can do with mouse and all, but I'll show you in a simple way, but you can also read these things that will help you how to use mouse in SAMSON, because that is different than other molecular visualization tools. Okay, so now I have the structure imported into my SAMSON software, so I can see the structure. I can see a lot of other rendering is done on this one. Now, as you're doing something in SAMSON, you have to create a SAMSON kind of a document, or it's a session that you are doing in SAMSON. So SAMSON calls it as a document. So how you can do it? You first have to, whenever you are importing any structure or doing anything, you just have to create a document from File, you go to new and create document. In this case, you can, I'm just creating a LAS R because this is the name of the protein I'm working with, and you can just customize this according to your needs. I'm going to press OK, and you can see I have that particular document. And then you should be working everything in this particular document because it's easier for you to save that document. So now I'm in LAS R document, because in this case, what will happen, you can work on multiple documents, but one document will be activated at one time in this case. Okay, so next is I'll redo what I've done so that you understand how the protein structure is imported, or we can get the structure by using this fetch extension and just type 4UV0, which is the protein PDB ID, and just click on the load, and then you will get the structure directly, and you can just close this, simply close this. And there are other options also. I forgot to mention that you can go for AlphaFold also, AlphaFold database, where if you don't have the crystal structure of the protein, you can directly go to AlphaFold. So there is no need to go and download and all to various databases. Everything is available. You can get the file in PDB, PDBX, and also MMTF file format. Everything is available. I'll just close this one. You can see these are the details that you'll get, and once it's done, you're good to go. Now, as you can see, you have the protein structure. You have these dotted molecules, water molecules that we all know, right? And now let's play around with the mouse. So in mouse, we have left click, right click, and then the center one, right? So what I can do with the left one and the scroll button, if you want to zoom in, zoom out. So using scroll, you can zoom in and zoom out. Pretty simple. By using the right click button, what you can do is just right click and do not leave it. Then you can move the structure like this side and that side. So I'm not leaving my right click button. I'm just pressing it down, keeping it in the same position, and then I'm not leaving it, because if I just click on the right click mouse button, I'll get the options that are available to me like this, right? So you have to click on the right click, and then you can move the structure up and down like this. You can just visualize. So right click mouse button is more important in this case. Left click if you want to select something. So if I want to select the entire protein like this, I can just do this, and my protein is selected. And you can play with select in a different way. But I'm just showing you with left click, you can just select the entire protein. With the right click, you will just move it. With the scroll, you can zoom in, zoom out, right? Okay, so as you can see, structure is pretty amazing. Visualization is really good in SAMSON. And then you have this tool which will help you to also move your structure. So if you just click on this one, whatever orientation you want, you can just move this entire cube in various orientations, and then your structure is basically going to move in this in that orientation. If you want this view, if you want the side view, it's going to move like that, right? And then you also have this nice scale bar which is provided with the SAMSON interface, right? There are various options right over here. Next is, we usually, when we work with any molecular visualization tool, we want to have a background of our choice. So in this case, as you can see, this toolbar which is available on the bottom, you can do zoom in, zoom out from here also. You can click on this one, zoom out. You can click on this, and yeah, okay. So this is not zoom in, zoom out. The second, this plus and minus is zoom in, zoom out, and this will help you to keep your structure in the center. Sometimes we get the structure and we lost the structure, so this will help you to get the structure in the center. And what you can do next is, by using the scroll, I told you that you can zoom in, zoom out, but by pressing the scroll, you can move entire protein structure like this. Basically, if you want to get hold on to this protein structure, so you just press and move it like this. Okay, and then I was telling you about the background, right? So this is the option that is available for the background. You just click on this one, and you can change it to white. Most of the academic work you want to publish it, you need a white background. You can do it black background because it is going to help you if you are working too much. But in my recommendation, is too dark of a background is also not good for your eyes, right? And then you have the option of custom. That means you can also create different types of background. In this case, go to settings. If you go to settings, and then you will get the option of choosing different colors, right? So if you want to have the gradient, so you can change the first color differently. So I can select this one, and then you can change the bottom color also. I don't want the gradient, so I'll just keep the color same, and I'll save it, and you can see this is the color that I have. You can obviously change this, right? You can make it default. It will be like this. There is another option that is available right over here after again the background option. You have this cube option, and then after that you have this this kind of platform option, which is also really good, because then you know how to visualize this protein, which is the top and the bottom of this particular protein, right? So it's a kind of a becomes a Blender's interface. So you can actually see what you're doing. Otherwise, you can be visualizing your protein upside down. So this tells you that this is the stable orientation. This is the right orientation, and then you can move along, because you will be watching lot of structures. You will be watching ligand molecules, so you'll obviously move. If you're interested in that one, you can just keep it, or you just by clicking this, it will go away. It's up to you, right? And then there are other options. We will explore these options, but let's do something else with this particular protein. So now we'll go to visualization now, and I'll show you how you can change the visualization of your protein structure. You have the option of labels. Okay, so just observe carefully when I do anything in SAMSON. Here I will have those specific tasks that are performed on the protein, and then you can just keep them by clicking on this one, or just you click on this and you can hide them. So as you can see, the receptor is by default shown as the ribbons in this case. If I'm clicking, then the ribbon is, if this is tick sign, then I can see the ribbon. If that is not, it's going to go out. Now, if I want to label this, you can add the label to any of these things that you want. Let's say you want to add the label to the molecule. So you know that this is for UV0, and then it's in the center. It's not going to move. It will, separately. When you move the structure, it will move along with the structure. And then you can see suddenly this appeared, and I can hide this, right? And you can keep this if you want, and then you can just uncheck this button. It will be hidden. The same way you can do a lot of things. I can label all the residues. Obviously, it's going to be very, very crowded, but you can see over here now, and I'm going to make a separate video on labeling and how we can do like a publication quality labeling in SAMSON. But you can see residues are labeled everywhere, and I can see a residue label, and I can click on this one. Residue labels, they are called, but I have that attribute right over here. Now, same way you can label everything right over here, even bond, even lengths, even atoms. You can do a lot. Now let's move on to the color. If you want to have different color, they are available to you. You can also use preset colors. This is the preset color. You can per element you can also do it. Look at this. So it's going to change based on the backbone. It got changed, right? Constant illustrative. Okay, so you can also provide a specific color to this. I don't know, let's use this, and yeah, you can actually see that has been applied, but there is no effect on these shadows and all, which looks good, right? Okay, what else? You can also do custom per attribute. You can also based on the chain, because it is having only one chain, and partial charge also, right? Okay, there are so many things you can do, and the structure is looking pretty good. Okay, what else? Visual models. So you already have visual models like this. So let's do the ball and stick. So this is ball and stick model. Okay, and I'll move on to illustrative, I guess. Or let's preset color, right? Yeah, because then we have multiple colors to different chains. Okay, so this is ball and stick, and I can see ball and stick. So let's hide the ribbon one, and you can see ball and stick clearly. And if you want to just show the ribbon and then hide the ball and stick. Now let's visualize literacy, and then you can hide the receptor. And this is when you have, another visualization in this case, which is literacy. Next is line. I'm clicking on line. You are not able to see this because literacy is also there. Now you can also see the line representation. Look at this, how easy this is, how convenient this is to change everything by just clicking on that, and quickly changes the visualization in a very, very interesting way. Now Wonder Walls, so you'll get the sphere representation, and you can see the visual appearance of the molecule is pretty amazing. Now I'll hide this, and if I hide the Wonder Wall also, everything is going to be hidden except those water molecules, because I have not excluded those. So this is Wonder Wall, which is again preset cartoon representation. Now for that, obviously you need to hide the van der Waal because that is the bigger side. And look, in this case, you have this is the cartoon representation. And then let's move on to the next one, which is ribbons. And then for that, you have to hide the cartoon representation, cartoon 1. And then let's move on to so many things, right? And all is getting saved. And then you just have to check them and then they will appear immediately. Next, what you have is Gaussian surface also, or tubes. If you want to show tube representation, cartoon plus tube looks pretty interesting, isn't it? Never seen these kind of images, but looks really good. Okay, so let's hide the ribbon representation, and this is just the tube representation. And you will see those kind of representation all the time in books and research papers everywhere. And then you have Gaussian surface also. Let's hide this and let's visualize Gaussian surface. See how much fun this is, and this is basically the surface view, right? How much fun this is to play around with the structure. How much easy this is. There are so many options that are available. I really like, playing with the different attributes. So I'll do the custom. Yeah, you can do a lot of customization on the color aspects. So let's do the custom one, and then this is default palette, HPV palette. I don't know. Yeah, you guys please explore this one, and lot of features that are available. So this is for the surface, Gaussian surface visualization. Next model that you have is solvent accessible surface. So it will tell you that this is the surface which is available or accessible for the solvent. And next, let's see the visualization. Visual mode is excluded surface. Okay. Yeah. So that is which is excluded by the solvent. Pretty interesting visualization. Okay, I'll hide this one. And you also have visual presets, which is illustrative. Look at this. These are already been given by SAMSON. So these are preset visuals. One is this, illustrative, protein ligand. Okay, I have to hide the first one. This is protein and ligand. You can clearly see the ligand molecule. If I zoom in this case, you can see the binding pocket. Wow. So it's also telling you the ligand binding sites also. And then if you click on this one, it will tell you which amino acid is this. Interesting. So in one go, you can understand where is the ligand binding pocket, what are the amino acids. If I do the label, and it will be immediately labeled. Anyways, that I'll keep for the next video, right? Okay, protein ligand binding site with Gaussian. All right. So this is telling you, and let me hide this one. Let's see what happens. Okay, it will remain like this, but it will tell you the surface area, and it will show you the binding pocket of the ligand molecule, right? And then you can use this box to move around. But yeah, obviously take your time to move the molecule, but you can see ligand molecule is sitting in the ligand binding pocket very nicely, and you can see this by using visual preset. Okay, so I think we have covered a lot about visualization, how to see protein structure or any structure in different mode, how we can use mouse control, how we can see various important features like how to get the structure in SAMSON, and then how all those attributes you can visualize them differently. Okay, so now let's save everything. So how you can save it? You can just press Ctrl S, and you can also save. If you want to have the image saved, you can save the image from here. Save image as. You can also click in a different format. You can save the image into PNG, JPEG, right? You have to provide the name. Okay, so by default, it's going to give you a specific address. You can change the address and you can save the image. But before that, I want to save this document. So you will go. So basically, this is very important to save the file or the document. So we'll save the document first. Save the document. We press Ctrl S, and as you can see, I've already directed this to my computer to a specific folder. You can also direct to the same folder, and I'll type this as last R01 session, and then I'll save it. Or let me show you how you can change this address also. So for example, right now I can go to my computers, right? And you can go to this, you can go to wherever you want to address this, and you can also go to download, you can go to desktop, you want to save this, right? So let me save this to data folder, somewhere data folder outside. So I'll save it. Now if I go back to my system, if I go to my system, where I'll go to data, and a lot of data files I have, and inside that you should be able to see this, right? Last R01 SAM .sam. This is the SAMSON document. What you can do is, now again, if you want to check this, you can go to SAMSON, open the SAMSON again, and when you do that, it will obviously take time because I do have extensions, and now it will take few seconds, and then the entire interface of the SAMSON will be available to you. And in this case, I can reopen that document. So I'll do open, I'll click on open, I'll go to my computer data, and then there I have last R SAMSON file. Click on this one, and then I should have the SAMSON file saved as last 01. All right, so that was all for the introductory video on SAMSON. I hope that you were able to understand and also able to follow along and seen by yourself what are the different modes that are available. I'll be making more videos on the SAMSON, and I'll be frequently using this tool for my research also, because a lot of visualizations that are really new for me, and I can see there are various things that I can do in SAMSON. So I hope you will also have the idea of using SAMSON for your research also. Please do let me know if you have any doubts, and let me know if you want a specific session on specific type of feature that is available or function that is available on SAMSON. I'll try to explore more of the SAMSON, and then I'll create videos on that one. So you will be seeing more videos on SAMSON later on my YouTube channel. So if you're new to the channel, then please do subscribe to the channel, and I'll see you in the next one. Till then, take care.